Most of us have heard of this term mutations before, but we need to give it a proper scientific definition. So we will define the term mutation as a permanent change in the order of the bases in DNA. Now, there are several things that can cause mutations. We've already discussed one of them, errors that get made during DNA replication. These create changes in the order of the bases in those new DNA strands. Remember, most of them are going to get corrected, but some of them will not get corrected, and they will persist and become permanent. They will become mutations. Exposure to certain chemicals, of course, can also mutate DNA. We refer to these as carcinogens as well because the mutations that these chemicals create can lead to cancers. And finally, exposure to certain forms of radiation can cause mutations. It's important to remember that in the genetic code, the codons, the groups of 3 bases that are used to code for one amino acid, have some redundancy built in. In other words, we said that for each amino acid, there are at least 2 codons that code for it. Some amino acids are coded by 4 codons. So some mutations can occur without changing the code, without changing what amino acid gets placed where. Other mutations are not quite as safe. Other mutations cause changes in which amino acid gets placed where. And of course, if you alter the positioning of the amino acids, you're going to alter the protein that is being made. So, all mutations change the order of the bases, and that means they're going to change the codons, the groups of 3 bases that get read as the genetic code. That means that there is always a potential to change the position of amino acids in a protein. It depends on where the mutation lands. If a mutation changes the codon in a way that changes the amino acid that it codes for, that's going to change the protein that's being made, and that's going to change the trait that comes from that protein. So when we discuss mutations, we categorize them because some mutations are structurally different from others, and some mutations have more of a consequence to the organism than others. Remember, all mutations change the order of the bases. All mutations change at least one codon, but that doesn't mean necessarily that there'll be a change in the protein. But if there is a change in the protein, there's going to be a change in the trait. All right, let's take a look at these categories of mutations. There are really only 2. There are what we call point mutations, and you may have learned about point mutations at some point in your academic career. These are the kinds of mutations where one base is out of line. One base is incorrect. Then there's a whole group of mutations called chromosome level mutations, and you may not be aware of these. It's possible for entire sections of chromosomes to break off and to sometimes get reattached incorrectly. Sometimes they don't get reattached at all. As you can imagine, chromosome level mutations are extremely serious. And yes, chromosome level mutations do happen in higher organisms like human beings. We're going to focus on the point mutations though, because these are the ones we see in our bacterial and archaeal cells most often. There are 4 kinds of point mutations. The first one is called substitution. The second one is silent. The third one is nonsense, and that's on the next slide. And the fourth one is frame shift. Let's talk about how these mutations differ. A substitution mutation, like the name suggests, is simply where one base has been substituted for another. So, for example, if there's a T in the old strand of DNA, the enzyme should put an A in the new strand. If it puts something other than an A, you have a substitution mutation. When a substitution mutation occurs, it can lead to a change in which amino acid gets placed at that point. And if that happens, it can change the protein and, of course, then change the trait. Silent mutations are one type of substitution mutations in that in a silent mutation, we've got a base being switched for another base. The difference between a silent mutation and a substitution mutation is the consequence. When a silent mutation occurs, the change in the order of the bases does not change the amino acid that gets placed at that location. And that means it doesn't change the protein at all, so it doesn't change the trait. We call them silent mutations because you would never know they were there. The gene is still making the same protein and giving the same trait to the organism. So even though there's a mutation, there's a change in the order of the bases, there is no consequence to the organism for that change. Nonsense mutations also involve one base being swapped out for another. The difference with nonsense mutations is that there is a severe consequence to the organism. What happens during a nonsense mutation is that a codon gets mutated to become a stop codon. Remember what a stop codon is. It's a signal to the ribosome to stop translating at that point. Let's imagine that there's a gene that codes for a protein, and this particular gene has 100 codons in it. When we look at those 100 codons, each codon being a group of 3 bases, that means we're going to have 100 amino acids in our protein. There's going to be a start codon at the beginning, and there's going to be a stop codon at the end. When the ribosome puts the 100th amino acid in place, it will then move forward and it will hit the stop codon and it will stop translating. Now imagine if somewhere like at the 20th codon, a mutation occurs. And the mutation changes the 20th codon into a stop codon. Now what's going to happen is the ribosome is going to begin translating at the start codon. It's going to put an amino acid in for codon 2 and 3 and 4 and 5. But when it gets to codon 20, it's going to hit a stop codon and it's going to stop translating. We sometimes refer to this as a premature stop codon. And as you can imagine, it has very severe consequences because depending on where that mutation is, the resulting protein can be much shorter. In my example, we're supposed to build a protein with 100 amino acids, but we're building a protein now that only has 20 in it. So as you can imagine, we often refer to these as nonsense mutations because the protein that results can sometimes be just pure nonsense. It's nothing close to what it's supposed to be. The last kind of point mutation we need to be familiar with is called a frame shift mutation. In a frame shift mutation, instead of substituting one base for another, what happens is an extra base gets added in, or a base gets removed. So you end up with new DNA that either has one less base than it should, or one more base than it should. And if you try to imagine this in your mind's eye, if I add in an extra base or I take out a base, what's going to happen past that point is that all my groups of 3 bases, all my codons, are going to get shifted by one base. If I have a set of 3 bases and then another set of 3 bases and then another set of 3 bases, I have those codons in place. If I add an extra base in somewhere or take a base out, all my codons get shifted over by one base. So everything past the point of mutation is changed. All of the amino acids, potentially, past the point of mutation could be different. And I could get a massively different protein as a result. So, as you can see, even with point mutations, even when one little base is put in the wrong place, there can be very severe consequences for the organism. Even in human beings, we have examples of terrible diseases that are the result of a single point mutation in the human DNA. What I'd like to do now is switch gears and leave the topic of DNA replication and mutations and take a few minutes to talk about a very important technique in microbiology called polymerase chain reaction. This is a technique that is important across all of the life sciences now. It's a commercially available technique. It's relatively inexpensive, and it has brought our abilities in medicine and healthcare forward quite a bit over the past couple of decades. In fact, the test that's being used today to test for the presence of the coronavirus is a PCR test. So, what PCR is essentially a method that allows us to do DNA replication in a test tube. It's going to allow us to mimic what a cell does and copy some DNA. Not only are we going to copy it, but we're going to copy it a lot. We're going to make so many copies that we can see them in the laboratory, and we can do certain things with them. So we describe PCR as being DNA replication performed in vitro. In vitro. If you're not familiar with that expression, that means in glass. It's a phrase we use to describe work that's being done outside of a living organism, in vitro. Now, what we do in PCR is we have a region of DNA that we're interested in. Usually, it's a gene. And we're going to use our in vitro DNA replication system to make copies of this gene of interest over and over and over and over again. We describe this as amplifying the gene. We're going to make so many copies, we're actually going to make millions of copies of this one piece of DNA, this gene that we're interested in. And again, the reason that you would want millions of copies of a gene is so you can actually do something with that gene in the laboratory. Now, PCR requires a very particular type of DNA polymerase in order to work. It's a type of DNA polymerase called Taq polymerase, TAQ. And it's a polymerase enzyme that's going to withstand some of the conditions that we need to perform DNA replication in the laboratory, and particularly the condition of high heat. Taq polymerase comes out of a living microbe, and it's a microbe that lives in heat vents at the bottom of the ocean. So this particular form of DNA polymerase is adapted to working in very high heat. And again, we use it because when we do PCR in the laboratory, there are times during the reaction that the reaction tube gets very, very hot. This picture over here on the right side of the slide, this is a picture of the machinery that we use to do PCR. This is called a thermocycler. And in 2020, thermocyclers cost a couple of $1,000s each. They are relatively small, about the size of a small microwave, and they're programmable. So essentially you can set up a PCR reaction and you can put it into the machine and turn the machine on, and you don't have to come back for several hours. And when you do, you'll have your millions of copies of your gene. So let's talk about the PCR reaction. We're going to use or take advantage of the DNA replication that occurs naturally in a PCR reaction. It's going to take advantage of the complementary nature of DNA replication. And because we're doing it outside of the organism, in vitro, we're not going to have all those great enzymes available to do all the steps of opening up the double helix and building a primer and so on and so on. Instead, we're going to use temperature changes to do a lot of these things, and we're going to use pre-formed primers. So during step 1, which we call denaturation, we're going to put our DNA in the tube and we're going to heat it up to a very high temperature, 94 degrees centigrade. And that high heat is going to break the hydrogen bonds between the two strands of old DNA. In other words, it's going to denature the DNA. Step 2 is called primer annealing. And in this step, we bring the temperature back down. It's still going to be pretty warm in there. It's going to be about 54 degrees centigrade. But this is a temperature that's going to allow pre-formed, pre-made primers to simply anneal with or bind with the old DNA. Wherever there's an A in the old DNA, there'll be a T in the primer. If there's a G in the old DNA, there'll be a C in the primer. So we design these primers to bind to the very beginning parts of this gene that we're interested in, so that the polymerase enzyme can come in and copy the gene. Now, we have to put primers into a PCR reaction in pairs. Remember, there are 2 strands of DNA, so we need 1 primer for each of the strands that we're copying, so the enzyme can come in and build new DNA on each strand. During step 3 of the PCR, we're going to heat the reaction up again to about 72 degrees centigrade. This is the step called extension, and this is the time during the PCR that the polymerase enzyme will make the new DNA off of the old DNA strands. At the end of that time, we're going to cycle or repeat the entire process over again. So if we put regular old DNA polymerase into a PCR reaction, into a thermocycler, and we heated that reaction tube up to 94 degrees centigrade, that DNA polymerase enzyme would fall apart and it wouldn't function. Ordinary DNA polymerase enzyme cannot withstand that kind of heat. So when PCR was being developed, the researchers went out searching for an organism that lives in that kind of heat, because they knew that their DNA polymerase enzyme would be able to withstand it. That's where Taq polymerase came from. There's an organism called Thermus aquaticus that lives in big thermal vents on the ocean floor. And what we've done is we have grown Thermus aquaticus in the lab, and we have isolated what we call Taq polymerase from this organism. And this is the enzyme that we do PCR with. Now, this diagram is very busy. Let's walk through it together so that we can see some of the dynamics of PCR. Up here at the top, they're showing us the stages that we just talked about in the PCR reaction. Here's our double strand of DNA. During step 1, we're going to denature. We're going to separate the two strands. During step 2, we're going to bring the heat down a little bit, and we're going to have annealing. The primers, the RNA primers, are going to come in and bind. During step 3, we're going to warm it up a little bit, and we're going to have extension. The Taq polymerase enzyme is going to come in and build DNA on one strand and on the other strand. So now, instead of having one double-stranded molecule of DNA, now we've got two. Now, I told you that the beauty of PCR is that you can get millions of copies of this piece of DNA. And that's really where PCR is so amazing. That's what this second diagram is about. It shows you that if you cycle the reaction, in other words, if you do denature, anneal, extension, and then do it again, denature, anneal, extension, and then do it again, upwards of 30 and 40 times, you get millions of copies of the DNA made. I know that seems hard to believe, but look at this diagram down below and you'll see this exponential amplification of this little piece of DNA. Here's the original DNA we started with, and they're showing us that our gene is in red. This is the piece that we're really interested in. We're going to go through one cycle of PCR. We're going to go through denature, annealing, extension. Now I've got two copies. Now I'm going to do it again. Denature, anneal, extend. Now I've got 4 copies. From 4, we're going to go to 8. From 8, we're going to go to 16. From 16 to 32, from 32 to 64, to 128, to 256, to 512, to 1024, and so on, and so on, and so on. If you get out to 30 to 40 cycles, you're going to end up with millions of copies of your gene of interest. That's the end of our slide set for the DNA Replication and Mutations lecture. You are ready now to take the online quiz.